I'm very honored to have this opportunity to present our work to you today. So this paper was a collaboration with Qianqian Permanente, Northern California. So our goal for this study is to identify genes that are associated with breast cancer survival. Those genes may be potential targets for treatment or clinically useful biomarkers for risk stratification and outcome prediction. Before our study, both animal and human studies support genetic influence on breast cancer prognosis and outcomes. There were already 11 genome-wide association studies on breast cancer prognosis that were reported. 24 genomic loci have been identified and 4 of them reach genome-wide significance. However, the problem is none of those loci overlaps across studies. Additionally, findings from candidate gene studies are inconsistent and lack validation. Therefore, we decided to carry out our own genome-wide association study on breast cancer survival. The genome-wide association study, also called GWAS, tests associations between the genotypes of genetic variants and phenotypes of interest. Study participants are usually genotyped using commercial genotyping arrays, which interrogate millions of genetic variants, also called single nucleotide polymorphisms. A p-value less than 5 × 10⁻⁸ is required for genome-wide significance in order to control for multiple hypothesis testing. Our study population is the pathway study, which is one of the largest prospective breast cancer survival cohort with extensive clinical and treatment information. This study includes more than 4,500 newly diagnosed female breast cancer patients. The pathway studies include patients from 4 major populations: European, East Asian, Hispanic, and African. We performed association tests between SNP genotypes and overall survival, while controlling for covariates like age, BMI, ER/PR HER2 status, treatment status, stage grade, as well as population structure within each population. And then we carry out meta-analysis across the 4 populations. This graph shows our final result. The x-axis is the chromosome position, and the y-axis is the minus log 10 p-value. Each point corresponds to a SNP, and the red horizontal line corresponds to the genome-wide significance cutoff. So we found a locus in chromosome 15 almost reached genome-wide significance. This locus contained a gene called UACA, which encodes a key regulator of tumor suppressor prostate apoptosis response 4. PAR4 recently received a lot of attention because it can induce tumor-specific apoptosis. The cancer cells, oops, what did I do? The cancer cells have PAR4 receptor GRP78 on the cell membrane. Binding of extracellular PAR4 to its receptor on the cancer cell membrane induce downstream apoptosis, while normal cells have little or no PAR4 receptor on the cell membrane. That's why normal cells are immune to PAR4-induced apoptosis. A recent study found UACA modifies extracellular PAR4-induced apoptosis. Usually in cancer cells, PAR4 bind with its receptor GRP78 in ER to escort it to the cell membrane. UACA interact with PAR4 in the ER to sequester it in the ER so that its receptor can no longer be transferred to the cell membrane. This results in less PAR4 receptor on the cancer cell membrane, and therefore the cancer cell become resistant to PAR4-induced apoptosis. Multiple recent studies reported PAR4 inhibition is required for breast cancer recurrence and resistant to chemotherapy. Consistent with these reports, we found chemotherapy significantly modifying genetic effect of the UACA locus in the pathway study. Additionally, a recent study found that HER2 target therapy and doxorubicin treatment results in PAR4 activation and lead to cancer cell apoptosis, while the cancer cells that have low PAR4 expression become resistant to those treatments and become the seeds for breast cancer recurrence. So we defined patients receive PAR4 dependent chemotherapy as those who were HER2 positive and receive HER2 target therapy, as well as patients receive doxorubicin. We found that PAR4 dependent chemotherapies have significant interaction with the SNP genotype in the UACA locus for overall survival, while other chemotherapy have no interaction effect with the SNP genotype. And then in light of this, we stratify patients by whether or not they receive these PAR4 dependent chemotherapy. And we found very strong association with overall survival and the UACA locus in patients who receive PAR4 dependent chemotherapy, while no association was observed in this locus in patients who didn't receive those therapies. It's also worth noting that the genetic effect is consistent across all 4 populations. Here is the zoom-in plot for that particular locus. So the x-axis is the genetic position, and the y-axis is the minus log 10 p-value. We can see very strong association across the entire UACA gene. And this is our top SNP in the European population. And here shows the Kaplan-Meier curve for our top SNP in patients who received PAR4 dependent therapy and patients who didn't. So in this particular patient group, we found, so the red is patient who have CC genotype, the green and blue correspond to the CT and TT genotype. So patients who carry more T alleles have worse survival. And then we try to replicate our findings in additional cohorts. I only have time to show you one of our efforts using breast cancer patients from Shanghai Breast Cancer Survivor Study and Shanghai Breast Cancer Study. So this population is quite different from the pathway study. Unlike pathway study, which is mainly whites, this patient population is all Chinese. They didn't receive HER2 target therapy, nor did they receive doxorubicin treatment. The anthracycline agency they received are predominantly epirubicin and pirarubicin. Consistently, we found our top SNP significantly associated with overall survival in patients who receive anthracyclines, and no association was observed in patients who didn't receive anthracyclines. So the Kaplan-Meier curves, again, are consistent with what we found in pathway study. So this replication result suggests that the UACA affects overall survival in both European and East Asian populations. And UACA's effect extends to all anthracycline treatments. This graph summarizes the association of our top SNPs with overall survival across all the cohorts we studied. So the point corresponds to the effect size, which is log hazard ratio, and the line corresponds to the standard error. So at the end, we found this association is highly significant, and the combined hazard ratio is quite large, about 1.8. So in summary, we carry out a GWAS of overall survival in the pathway study and identifies a novel locus containing this UACA gene. This association is strongest among those who receive PAR4 dependent chemotherapy, which include anthracyclines and HER2 target therapy. Our findings are consistent with the UACA's role in inhibition of PAR4 induced tumor apoptosis. Our meta-analysis across pathways and 3 other prospective breast cancer cohorts reveal an even more significant association. We also perform additional analysis, which I don't have time to show you here, to support that UACA is likely the causal gene of the GWAS association, and higher UACA expression associated with worse survival. So at the end, our findings opened the door to a clinical test in predicting treatment outcomes with the 2 most popular therapies, enzocyclines and HER2 target therapy, as well as highlight the importance of targeting PAR4 pathway for improving breast cancer treatment. Last but not the least, I want to thank my collaborators, as well as the funding agency, particularly Dr. Song Yao and Dr. Larry Kushi, who co-lead the project with me. Thank you for your attention.